cell death on aging and chronic diseases. We're going to describe C15 or pentadecanoic acid and why it meets the criteria of being an essential fatty acid and the mechanisms of action relevant to metabolic, heart, and liver health. We're then going to help you understand the C15's evolutionary role in supporting mammalian longevity, thanks to dolphins, providing some insights, and how the cellular stability hypothesis explains how C15 can extend longevity not only across species, mammalian species, but individuals. We'll then describe cellular fragility syndrome, which we just, Dr. Melvin, as you just shared, we just published less than 2 weeks ago, which is the first nutritional deficiency syndrome discovered in 75 years, and how this deficiency syndrome accelerates aging and the onset and progression of metabolic liver and cardiovascular diseases. And then finally, to understand why we have nutritional C15 deficiencies affecting, we believe, as many as 1 in 3 people globally, how to test for C15 to identify low, healthy, and optimal levels, and most importantly, how to fix them. We'll talk about some dietary options as well as supplements as well. So with that, let's get into story mode, right? So if we look at our health and the health of your patients, I think we all know that our health is heading in the wrong direction. So despite all these advancements in medicine, a lot of effort being spent on how can we live to 180, 200 years old, which is all wonderful and very exciting, but what's happening in the real world is, our longevity is decreasing. So something is happening that's putting us in the wrong direction. And we've been starting to see over the past 10, 15 years, aging-associated diseases increasing, especially among younger people. And when I say younger, people less than 50. So at 52, I'm conveniently putting myself on the better side, but something has been happening, people younger than 50. Specifically, we are seeing an increase in coronary heart disease among people 18 to 44 years old. For a long time, right? For decades, we were seeing a decrease in heart disease in people. And suddenly around 2017, 18, increases specifically, again, among this younger age set. The incidence of type 2 diabetes, specifically less than people 20 years old, has been increasing exponentially. You remember type 2 diabetes used to be called adult onset diabetes, right? And type 1 diabetes was juvenile onset diabetes. It used to be really rare for a child, a youth, somebody who was young to get type 2 diabetes. We're now seeing that increase. That amount is expected to increase by 673% in this age group by the year 2030. Not only for the type 2 diabetes, not only is it showing up, but and growing, but it's more aggressive. So they're actually finding that these younger people that are getting type 2 diabetes have a higher risk of death than even children with type 1 diabetes. So it's a very aggressive form. And then non-alcoholic fatty liver disease, which of course is undergoing a name change, but for this sake of this presentation, I'm going to keep calling it NAFLD versus MASLD. But NAFLD showed up out of nowhere. In 1980, the Mayo Clinic published a paper in which they reported 20 individuals who had this strange presentation of fatty liver disease that mimicked alcoholic liver disease, but they were confident that this risk factor was not due to alcohol consumption. So they called it non-alcoholic steatohepatitis, or NASH, and those were the first 20 to 30 patients in 1980. Fast forward to today, and now 1 in 3 people globally have non-alcoholic fatty liver disease, the precursor to NASH, including 1 in 10 children. So it's pretty shocking how much this condition has increased exponentially. We're talking about, just as they go to review the global data, and published the next paper on the trends, it's gone from 1 in 5 to 1 in 4 to 1 in 3 with each subsequent publication. So then with among all of this, a new type of cell death emerged, and it's called ferroptosis. So, back in our cell biology days, we were taught there are 3 ways that our cells die, right? It's apoptosis, necrosis, and autophagy. And a team of scientists from Columbia University identified a 4th form of cell death, and they called it ferroptosis. And since it was their initial publication in 2012, there have been over 10,000 peer-reviewed scientific papers on ferroptosis. The level of detail with regard to the pathways and the genes is incredible over these 10,000 papers. Despite all this science, nobody has been able to explain why ferroptosis showed up and what is causing it. So it's been very difficult to figure out how to treat it. I'm just going to go in a little bit deeper on ferroptosis. It is a non-apoptotic form of cell death. This is how it is defined by the folks at Columbia University when they discovered it. It starts with fragile lipids, fragile fatty acids in the cell membranes. Right, our cell membranes are made of a lipid bilayer, and the fatty acids within those bilayers, if they are fragile, that leads to lipid peroxidation. Now, when we're talking about fragile fatty acids, these are fatty acids that have double bonds in them, and that every extra double bond they have increased risk of attack by oxygen and lipid peroxidation. So fatty acids that have double bonds are those that we call unsaturated fatty acids. And those that have multiple double bonds are called polyunsaturated fatty acids. So the more of those that we have in our cell membranes, the more fragile they get, the more susceptible they are to lipid peroxidation. So then this is the first part of it. The second part is that iron is strangely showing up inside of cells. That's free iron. And when that free iron then combines with lipid peroxidation, that results in massive reactive oxygen species creation. And as the ROS increases, that then attacks the mitochondria and results in the cell death. So when you look at the cell under the microscope, it is a slightly thickened cell membrane with a nucleus that looks just fine, so non-apoptotic, and but the mitochondria are dead. So, and there's iron inside. So it's a very distinct presentation that we see with ferroptosis. Oh, that was sweet. Okay, so and if there are questions, actually, Dr. Melvin or others, feel free to shout out as well during the talk. So from there, now we know, because of all of this work, right, that ferroptosis causes accelerated aging. And it's basically accelerating the process of aging-related degradation at the cellular level. It is also leading to earlier onset and more aggressive chronic conditions, including type 2 diabetes, cardiovascular disease, and NAFLD and NASH. So while all of this is happening, this feels so gloomy and doomy, but I promise we'll get hopeful here real quick. At the same time, especially in the US, our lifespans are decreasing. And what's important is that all of the things I'm talking about, this happened before COVID, right? So this is when around 2016, 17, 18, we started seeing these trends happening. And 2012, ferroptosis is discovered, and we were already seeing our life expectancy kind of plateau out and then head down. And then we got hit with COVID, which arguably we had super susceptible population that were more fragile to an added threat, right, which was COVID-19. So interestingly, so a lot of explanations with regard to these trends have been, with regard to diet and food, are increased sugar, increased trans fatty acids, too many ultra processed foods, and those, are probably reasonable contributors to what it is that we're seeing. But while all this was happening, I was in San Diego enjoying a stint with the Navy Dolphins. And this was 20 years ago, 20 some years ago. And I had been with this Navy dolphin population, and that's where introducing myself, right, as a veterinary epidemiologist, was invited by the Navy 20 years ago to apply veterinary epidemiology to understand chronic health in their dolphin population. So the Navy has cared for a population of about 100 bottlenose dolphins for over 60 years. They live in the open ocean in San Diego Bay. They go out in the ocean every day. Every day they choose to come back. It's really incredible population. Because they get such good care, Navy dolphins live a lot longer than dolphins in the wild. So a wild dolphin on average lives to 20 years. Dolphins at the Navy routinely live 40 to 50 years. So after I had been there for about, 5 or 6 years, the Navy has had this growing population of geriatric dolphins. And we started seeing an increase in a syndrome. And this included NAFLD and NASH, like phasic increases in liver enzymes, high cholesterol and triglycerides, insulin resistance, metabolic syndrome. We were seeing this dysmetabolic iron overload syndrome in which iron was depositing in the liver. They had hyperferritinemia, chronic inflammation, anemia, and even the full suite of changes in the brain consistent with Alzheimer's disease. So, and importantly, it wasn't all the dolphins were getting this syndrome, only a subset. And interestingly, about 1 in 3 were developing some component of this syndrome. So that then triggered us to, we were afforded the luxury, right, that we have a large-brained, long-lived mammalian population that are well-controlled. All they eat are fish. So we were able to go back into their archived serum because wonderfully, they collect blood on the dolphins as often as once a month and as rarely as once a quarter just as a routine health checkup. They measure 44 different clinical indices on every test. They have kept those blood records archived in electronic medical records since the 1990s and archived the serum of the dolphins and saved all the tissues that when dolphins passed on. So we were able then to come in to this amazingly rich resource and be able to use advanced metabolomics to be able to evaluate thousands of small molecules present in both the dolphin serum and in their all fish diet to figure out which small molecules might be predicting dolphins with or without this condition. And that's where we discovered, and to be honest, we went in with a biased approach where we just assumed it would be omega-3s, that higher omega-3s in their fish would predict the healthier aging dolphins. And instead we were proven wrong, and it was C15 that came out of this, of these initial studies, that dolphins with higher C15 in their blood had a lower risk of having these conditions, and as well as higher C15 in their diets. I guess important to call out here is that dolphins at the Navy are fed 5 different types of fish. 3 types of the fish were absent of C15, and 2 types had higher C15. Different dolphins were fed different proportions of these fish. So we accidentally had dolphins that were getting a higher C15 diet versus a C15 deficient diet. So while we were making these discoveries and publishing all of this work in the peer-reviewed literature about all these conditions in dolphins that were mimicking those in humans, as well as this discovery with C15, it ends up the same story was playing out in humans, that multiple studies, now meta-analyses, so studies of longitudinal cohort studies showing that people with lower C15, both dietary and circulating C15, have a higher risk of developing type 2 diabetes, cardiovascular disease, including especially coronary heart disease and heart failure, NAFLD, NASH, certain cancer types, and anemia. So all of a sudden it was like, there's what's happening here. And is this more than just association? So let's talk about C15. As shared, this is the first essential fatty acid to be discovered in 90 plus years. When we initially made that discovery of association in dolphins, we then moved pure free fatty acid C15 into the lab. And over 3 years, we did 8 studies, guided in part by Dr. Ed Dennis, who's a leader in fatty acids. He was the editor-in-chief of the Journal of Lipid Research for 15 years. And he said, gosh, here are the series of studies that we should look at to see if it's a beneficial an active fatty acid. And we found not only is C15 an active and beneficial saturated fat, but it was meeting these rare criteria of being essential, which means our bodies require adequate levels of C15 to maintain our physiological health, but our bodies can't make enough of it. That means that we have to get certain amounts of C15 from our diet. The dolphins needed adequate amounts of C15 from their fish. As we'll learn with people, we need to get adequate amount of C15 from our diet, and our primary source is whole dairy fat. So you can see C15 is such a simple molecule. It is 15 carbons that are all together, no double bonds, so it doesn't bend. So it's literally this tiny little 15 carbon chain, super simple. Nature has made this molecule essential nutrient to do a lot of really important things that we'll talk about. So it is a dietary odd chain saturated fatty acid. As I mentioned, it's a stable fatty acid with no double bonds. That means it's resistant to lipid peroxidation. And we'll talk about one of its most important points is it strengthens cell membranes. So we published this in Scientific Reports in 2020 and made the case that this was an essential fatty acid. So what the paper we just came out with is we've now taken the past 10 years of research that we've been doing on C15 and brought it all together along with the wealth of data from other folks around the world showing that C15 reverses ferroptosis and effectively stops all of the core components. We and others have been able to show that C15 gets into our cell membranes, it stabilizes and strengthens our cells by 80%, specifically in red blood cells. It prevents them from hemolyzing. It lowers lipid peroxidation. It lowers intracellular iron. We'll talk about that mechanism of action later. It has been shown to lower reactive oxygen species by 45%. And as a result, it repairs mitochondria and it restores healthy cells. So if you have a molecule that goes in and truly helps with each part of this process, this is now starting to point at, this is not just a fix, that having too low levels of it is likely driving the cause of ferroptosis. So in addition to ferroptosis, which then we again propose in our paper that this is the cause of ferroptosis, a primary is C15 deficiencies. C15 has multiple mechanisms to enhance health and longevity. So it is a pleiotropic molecule with these activities with broad effects. So this includes anti-inflammatory, antioxidant, antifibrotic, anti-cancer, and antimicrobial. We'll share in a little bit this suite of benefits all show in a dose-dependent, significant fashion in the peer-reviewed literature matches that of rapamycin. So we'll get to that in a little bit. So when we look at its specific receptors and mechanisms of action, it has these wonderful suite of longevity enhancing activities to the right by activating AMPK, inhibiting mTOR, which are key in the longevity pathway. mTOR obviously is the target of rapamycin. AMPK activation is the mechanism of metformin. In addition, we've been able to show that C15 activates PPAR alpha delta. These are known receptors, well known to help regulate metabolism and immunity, right? PPAR alpha activators are how fibrates work. to be able to decrease triglycerides and even LDL cholesterol. It's also an AKT activator, another win for glucose control, and it inhibits both HDAC6 and JAK-STAT. JAK-STAT is my favorite mechanism with regard to anti-inflammatory, broad anti-inflammatory mechanisms, and both HDAC6 and JAK-STAT have been demonstrated by others are the mechanisms of action that explain C15's anti-cancer properties. We have not been doing much in that area. A little bit, it's just whole other teams are working on that component and potential for C15. So when we move to beyond mechanisms of action, we then have how C15 has clinical efficacies that improve health. And this includes double-blinded and controlled clinical trials. When we look at the animal studies, C15 treatment, oral C15 provided over 12 weeks, effectively alleviates anemia, lowers liver enzymes, lowers cholesterol, glucose, alleviates liver iron deposition, and decreases liver fibrosis in animal models of type 2 diabetes, as well as fatty liver disease and NAFLD. Importantly, this holds true for a key model for ferroptosis, that is a rabbit model that C15 had remarkable abilities to reverse, just like we had seen in the dolphins. When we move to people, beyond the epidemiological studies, we now have 2 randomized controlled double-blinded clinical trials with pure free fatty acid C15 given orally over 12 weeks. One study was done in Singapore, and it was with women with a history of NAFLD, and the other is a study that is soon to be published. So excited for that to get out of young adults who were overweight and had a history of NAFLD as well. And what these studies showed was that a C15, oral C15 effectively raised hemoglobin. And again, we'll talk about these parallels, right? Of alleviating anemia, strengthening red blood cells, alleviating anemia in animals, raising hemoglobin, strengthening red blood cells in humans. Importantly, lowers liver enzymes, lowers LDL cholesterol, and improves the gut microbiome, which includes growth of a gut microbe called Bifidobacterium adolescentis, which is super fun because it's basically our core microbe that helps with regard to metabolic health. But it's also been shown to extend longevity in worms and flies. So might have a more interesting twist that it helps with the growth of these microbes. So have I mentioned? So C15 is the first essential fatty acid to be discovered in 90 years. Once we discovered that C15 was an essential fatty acid, we then wanted to test it head-to-head against pure omega-3, the highest performing, specifically EPA, and when we're talking about heart health. So we used this test this panel called Biomap, which basically looks at 146 different biomarkers across 12 human cell systems mimicking various disease states. So this allows you to test different pharmaceutical companies use this test routinely to assess not just safety, but efficacy for potential diseases. And so what we showed was that pure C15 had all of the benefits of pure EPA and then 3 times more. And additionally, it was safe. So because it has no double bonds, when it got to the higher doses of like 50 micromolars, it continued to have efficacy and be safe. Omega-3s, on the other hand, once it got to that 50 micromolar state, because of the lipid peroxidation, it actually started to kill cells. So an important part of attribute with regard to C15. we did the same test. So then we repeated it. As we started seeing that C15 was matching the benefits of rapamycin, we then worked with Dr. Nick Shork, who's the leader of the NIH's Longevity Consortium. And Nick said, this is looking like a geroprotector to me, and a great longevity enhancing molecule, especially with all the other molecules he is seeing going through the anti-aging pipeline. So we did the same test with Biomap, and we went head to head against a carbose, metformin, and rapamycin. And again, C15 had these 36 benefits, which was better, far better than acarbose and metformin, and was mighty close to rapamycin. So again, not a surprise that C15 and rapamycin had similarities based upon the fact that both of them have all of these activities on the right. So our conclusion from that paper, which we published in Nutrients late last year with Nick, was that rapamycin might actually be just a C15 mimic, right? And we didn't actually have to go to Easter Island to get this molecule that's made by a foreign bacteria and is very complicated and comes with a really long list of side effects. Instead, what we've really needed for longevity is C15, something that every mammal gets from birth in mother's milk. So it just has taken us a while to get there, and thanks to the dolphins to helping us discover it. So when we talk about we're moving into the longevity realm, that C15 targets 6 hallmarks of aging. The number keeps increasing the more the world has been working with it. Which shows that it slows aging at the cellular level. It slows cellular senescence by inhibiting mTOR and strengthening cells by 80%. We talked about it repairs mitochondria and lowers reactive oxygen species by 45%. It stops inflammation by lowering these 18 pro-inflammatory cytokines. It restores cellular signaling, all those important mechanisms of action that I talked about earlier. The new ones that came in were the gut microbiome, as I had just shared, and a really fun study showing that there's an association of C15 with slower biological aging, a study that showed that people with higher C15 levels had lower DNA methylation. And then when they applied it to these epigenetic clocks, they showed that the higher the C15 people had, the more likely their biological age was younger than their chronological age. So association, but we have a lot of reasons to believe why that C15 may actually be causing it versus just association. So the next piece with regard to longevity is talking about the cell membrane pacemaker theory of aging. And this was introduced by A.J. Holbert in 2005. And he came out with this theory where he showed the greater the cell membrane stability, the fatty acids in the cell membrane, the longer a mammalian species lived. So he was able to show by looking at about 40 or 50 different mammalian species that the more saturated the fats in the cell membrane, the longer a mammalian species lived. So we have much higher levels of sturdy saturated fats compared to mice, and then everything in between. So really exciting theory that matches the mechanism, a core mechanism of C15 with regard to ferroptosis and cellular stability. So what we introduced in this paper in Metabolites from a couple weeks ago is the cellular stability hypothesis, where we're taking that initial hypothesis theory and we're pushing it to say the greater the cell membrane stability, the longer an individual's longevity. So we believe that because of the evidence to date that we can push C15 levels, we need to get them at adequate levels, and that can help dictate the strength of our cell membranes and to help support, to stave off chronic diseases to extend our longevity. What we do is we do a very comprehensive review of all the studies to date on C15, and we tie that to our experimental studies over the past 10 years. And where they consistently land is that people who have less than, or dolphins that have less than or equal to 0.2% of C15 in their cell membranes, we're defining as having a nutritional deficiency, and they're undergoing accelerated aging. This is when cells become susceptible to ferroptosis. And it really feels like a threshold. So it's like literally you're standing at the edge of a cliff, right? If you're right at the middle of a 0.2% of C15. And if you take one step off and you lose that amount of C15, the cell membrane becomes fragile, ferroptosis. This is what we saw happen in the dolphins and as well as in the models. If we have greater than 0.2 to 0.4% of C15, then we're calling that nutritionally replete and that you are aging normally. Most people are standing on the cliff. So most people today are at 0.2% of C15 in their red blood cell membrane. Then we have encouraging science that shows from others that show that this going from getting to a 0.4 to 0.64% of C15 may actually be optimal, and that may actually slow aging better than what we have understood in the past. I'll provide the science on that front. So our C15 levels have been declining, and why, right? So it's because our primary source of C15 is from whole fat dairy products. And since 1977, right, we've been told to decrease our intake of whole dairy fats, specifically to decrease our intake of saturated fats, including C15. That has resulted in declining C15 levels. So back in 1950s, we drank an average of 2 cups of whole fat milk per day. And that is now down to about a half a cup of milk, not even whole fat milk. So non-fat milk has no C15 in it. So we've seen a dramatic decline. That has coincided with a decline in circulating C15 levels over time, where we've gone from a 0.23 down to this 0.2, and this was from 2011. So likely, like right around, if not below this level, is where really the world is now sitting. So it's not just because of decreased whole fat intake, but that's the primary driver. We now also know that how much C15 that is present in milk is changing, and that if a cow is fed corn, it has half as much C15 in its milk than a cow that is fed grass. So grass-fed, dairy products coming from grass-fed cows are better. And there are even more encouraging studies showing that animals, ruminants that are fed grass at higher altitudes, so mountainous grass, they have twice as much C15 as the sea level grass. So there are some actionable items in the industry that we can do to help get C15 levels back into our food supply. Additionally, our C15 levels go down as we age. So we're still trying to figure out why, if it's decreased absorption, eating less, but we do know that C15 levels go down as we age. Could that then be accelerating the aging-related breakdown? We argue likely. So this is a graph showing Sardinia, and we'll get back to Sardinia as well, but this is a high longevity zone. And Sardinia so Sardinia has some of the longest, like it's this blue longevity zone. It's where most men live beyond 100 more than anywhere else in the world. And it's been attributed to they have a lower risk of dying from heart disease. And so in Sardinia, they have, even our 60 to 75-year-olds have C15 levels that's 3 times higher than we see around the rest of the world of 0.6. Even the 80-year-olds, it's lower, but it's 0.4. And then what they called their reference low longevity zone, which is going to be us, apparently, is at that 0.2%. So we'll get back to that in a second. So then let's talk about cellular fragility syndrome. So what we share in this paper is that C15 deficiency causes cellular fragility syndrome. And again, this is the first nutritional deficiency described in 75 years since vitamin B12. So the way this plays out, the pathophysiology behind it, is that if we have less than 0 or equal to 0.2% C15 in our cell membranes, that makes us susceptible to cellular fragility syndrome. What happens is our red blood cell membranes become fragile, and this results in lipid peroxidation. When our red blood cells become fragile, then our macrophages in our liver, the Kupffer cells, then go to work, right? So they detect fragile red blood cells and they engulf those red blood cells, the weak ones, into the liver. And normally their job is to then just recycle the red blood cells and the iron. But if you have too many weak red blood cells and it's taking in a lot of those weak cells, that results in what's called dysmetabolic iron overload syndrome. And this is what we found was happening with the dolphins, that the red blood cells had become weak due to low C15. They were developing iron overload in the liver, and that resulted in visible iron and driving dysmetabolic iron overload syndrome. This then contributes to ferroptosis, that then has the downstream effects of first liver. So what's unique about this, and Jeff Schwimmer, who's a leader of NAFLD, pediatric NAFLD here at UC San Diego, he's like, I knew it. He's like, I knew it was all about the liver, that when we see the metabolic syndrome coming from this, it starts at the liver because of the injury that's happening due to ferroptosis in the liver, resulting in insulin resistance, hyperglycemia, dyslipidemia. NAFLD progresses to NASH. We get chronic inflammation. That then spills over, the iron spills over into the blood and then affects our other organs, leading to higher risk of direct damage to the heart, as well as in the brain. We aren't talking a lot about the brain. We'll have a lot more on that front next year. So cellular fragility syndrome, when you talk about how do you detect it in your patients, that there is now, and Genova Diagnostics offers an at-home C15 test that then allows, and it specifically measures C15 in the red blood cell membranes, which is exactly what you want. So having low, and they report it in percentage as well as in micrograms per mil. So you can find this amount. So that's the first part. Fragile red blood cells can be detected because you can still have fragile red blood cells that have normal red blood cell count as long as your production keeps up with the loss, right? So elevated red blood cell distribution width is a huge sign, not specific, but it is consistently part of this syndrome. Elevated reticulocytes and elevated osmotic fragility. I wish that test was more widely available. It directly tests the strength of the red blood cells, and you may or may not see low hemoglobin. Once it advances enough, that's when a low hemoglobin and anemia kicks in, and why we're arguing so many people who are older have anemia of unknown cause. This then, you can detect the iron overload component with hyperferritinemia, of course, ruling out chronic inflammation as the cause, but it's a hyperferritinemia that can have a normal total iron binding capacity. For ferroptosis, you can detect high lipid peroxidation products, specifically T-bars, and again, this high ferritin. And then once we move to the states where it's affecting the liver and the heart, then that's where we see the elevated glucose, cholesterol, triglycerides, liver enzymes, and specifically high IL-6, TNF-alpha, and MCP1. This is what the dolphins had. They didn't have any variation. Every dolphin had this picture. And so we were gifted with this. Not every person who has NAFLD has cellular fragility syndrome, and not every person who has cellular fragility syndrome develops these diseases. But as we get familiar with this mix of diagnostic tests, it may help with regard to seeing things that could be driven and caused by this disease. So that getting toward the end to be able to take questions, the big thing is that, this is fixable. The upside of a nutritional deficiency syndrome is we can detect it and we can fix it. So we now know that most people need an added 100 to 200 milligrams of C15 per day to not be deficient. Our bodies can make some C15, and that's specifically with microbes in our gut that can make some C15, but it doesn't appear to be enough. So most people, which is why most people are sitting right around that 0.2%. Some people who are strict vegans, we've seen levels as low as 0.04, 0.02%. So they would need more. But most people need to a conscious add of 100 to 200 milligrams of C15 per day. And what we've shown, importantly, is that adding C15 back, right, reverses cellular fragility syndrome. So we can replenish healthy C15 levels to restore long-term health. So if we can get up and over 0.2% of circulating C15, cellular stability comes back. We results in normal hemoglobin and normal red blood cell distribution. This is a remarkable fix. It happens within weeks. Where the anemia is alleviated within 3 to 6 months. We see a decrease. In fact, it stops a deposition of iron in the liver. Again, these are in relevant models. And we may or may not see lower ferritin. It's not helping to take the iron out that's already in the liver. It's just helping to stop it from accumulating further. It stops ferroptosis and all of the components of it. And at downstream in relevant models, and now in these clinical trials, we're seeing healthier liver, leading to lower glucose, lower triglycerides, lower liver enzymes, lower LDL cholesterol, and pro-inflammatory cytokines, and improved cognition. Again, I'm going to hold on that one. It's encouraging early data, but all these other areas have much, much more robust, healthy data to talk about patients. So optimizing C15 for longevity. So now saying, okay, now we can fix C15 levels and get back to where we're supposed to be. Is there a potential to go from that 0.4 to 0.64% to get to an optimal state? And so that's where we have one study by True et al., by folks at Stanford and other places, where we can see that the higher the C15, the higher the C15, a prospective cohort study, the lower their risk of developing cardiovascular disease. So if you have C15 greater than 0.4 to this kind of 0.55%, they had the lowest risk of cardiovascular disease. That matches with, again, this longevity blue zone of Sardinia, where they have lower risk of dying from heart disease, the longest lived male populations in the world, and they're sitting on this 0.4 to 0.6%. Why not just eat more dairy fat? Well, we could, and we argue that children should probably go back to being able to get whole dairy fat in their earliest stages. But for us, over decades and decades of life, dairy fat only contains 1%. Milk fat only contains 1% of C15, while we have it contains greater than 40% of pro-inflammatory fats, these even chain saturated fatty acids. There's a less bioavailable, it's in a less bioavailable triacylglyceride form, and it's highly variable C15 content based upon what I shared earlier. We don't know what's in that milk anymore because we don't know what the cows were fed, the season, the altitude in which they were fed grass. So that's where optimizing nature to extend healthy aging and longevity. How can we push science to make use of nature and be able to see if we can get to that 0.4 to 0.6%, not just for the folks living in Sardinia. And that's where a pure C15 free fatty acid may be the way forward, and based upon input from a panel of experts that met. And they said, when it comes to dairy fat, it does seem that the individual nutrients, ingredients within dairy fat do better on their own versus when provided as a whole. And this is an exception to the general rule, where it's better to get your nutrients by eating the whole foods. This group concluded that for dairy fat, that may not be the case. So with that, I will share, you can nerd out on the science. We've been winning all kinds of awards for our science. We're proud of it. And you can nerd out on all of the peer-reviewed literature at discoverc15.com. Whew, that was a lot of talking. I'll end it there.
That was great, Dr. Staff.
Thanks.
Thank you. Dr. Bill, you want us to read the questions?
Yes, go ahead.
Okay, so question here. Oh, Stefan's probably also a difference between raw and UHT pasteurization in quality and quantity of C15, same as lactoferrin and other enzymes that denature with heat.
So there've been a couple studies with pasteurized and raw milk, and I thought the same thing would hold true, but in those, it's only two studies, but in those two studies, they showed they had the same amount of C15 in them. More studies need to be done, but right now, it seems more of what happens to the cow as far as how it's fed versus what we do with the milk after.
Right, yeah. So the quality of the cows really are questionable now. I mean, steroids, antibiotics, hormones. I think that also plays a big role. Another question is C15 omega-7.
Yeah, it is not. So omega-7. So different. So omega fatty acids are polyunsaturated fatty acids. It's a good question. And so those are by default, if it's an omega fatty acid, it's going to have double bonds in it, while C15 is a saturated fat, which right goes against everything we've been taught for all this time. Ends up there are good saturated fats and bad saturated fats. C15 is a saturated fat, but has 15 carbons, an odd number versus an even number, which C16, C18, which continue to be associated with a higher risk of type 2 diabetes, heart disease, fatty liver disease. So it's kind of two camps, saturated and unsaturated. C15 is on the saturated, and then within there, on the odd chain fatty acid side, which is now indisputably healthy saturated fats.
So when you're saying saturated fat, that has a misnomer for everyone that they're thinking it's always unhealthy. But in this
Yeah.
situation, 15 kind of twists or turns the table around for the reputation of saturated fats. Is that correct?
Yeah, that is absolutely correct. And I think, a lot of people ask is like, how was this not discovered before? And there were studies, plenty of studies, epidemiological studies showing that people who had higher odd chain fatty acids, C15, C17, had a lower risk of developing these diseases. The problem was that these huge studies were done to show that saturated fats are bad for us. And it did show C16 and C14 and C18 were associated with a higher risk. So that's where the conclusions and the conversation focused. And the mention of C15 and C17 was almost like a postscript. And it's like, oh, and by the way, there were these trace odd chain fatty acids that were associated with a lower risk of diseases. But the big story is even chain saturated fats present in higher levels in dairy fats increased risk of diseases, check. So it really took the dolphins to be like, no, this is real and not just an interesting association.
Right. Okay. Does this imply vegans do not consume C15 at all?
That is a good question. So they probably eat very little C15. So there are some plants, like sunflower oil, plant-based products that has C15 in it. It is combining now with unsaturated fats, which can compete with, right, the benefit of this being a stable fatty acid. And when we eat fiber, one of the molecules, right, from fiber, one of the benefits of fiber is inulin. And a really cool study showed that inulin is used by our microbes to make C15. And it was because of that production of C15 specifically explained how fiber decreases the risk of fatty liver disease and NASH. So there is some production from plants, but what it appears to be is it's just not enough to keep us off or over that cliff.
Right. So as in vegans, as I see patients, I do panels of nutrient and mineral deficiencies. So now you're saying C15 can be assessed also as part of that mineral assessment. Maybe not only vegans, but also vegetarians. And you said there was a test also that's available out there that can be used to see this. I mean, you mentioned a specialty test, but regular testing, regular labs can do this for us, or no? Just a specialty lab that you just mentioned.
No, others can do it, but you need to look. So they need to offer a fatty acid panel. Ideally, what they're testing, because they could test in the serum, in the blood, or in the red blood cell or erythrocyte membrane test, right? So the ideal one is erythrocyte membrane, but it's pretty consistent among all three, serum, plasma, and erythrocyte membrane. So first check and see that the labs you typically use, if they have a fatty acid panel, and if they do, if it includes C15. So for a long time, C15 was not included in fatty acid panels, which is one reason why it was hidden for decades, because C15 used to be used as the reference molecule to measure all other fatty acids. So it never got counted, it never got evaluated as beneficial because it was the ruler instead of, so another reason why it was hidden for so long.
I
I
do
think,
remember, yeah, I do remember this in relation to asking for a panel, and yeah, now I should look into C15 if they include that.
yeah,
So okay,
and demand it. I mean, tell them if they have a fatty acid panel that does not have C15, encourage them to include it. So it's
right.
unique.
Yeah, from Beverly Food
Let me let
Ways.
me let
Oh,
me
go ahead, go
let
ahead.
me get something go. Let me get something through here, which is very maybe interesting for most of you. Um, the best indicator for health optimization, and we have a lecture on Monday, is endothelial function. So as long as the endothelial function is optimized, the patient's health is optimized. So that's a very good way of measuring the efficacy of supplements. If we try with patients and measure their endothelial function test and then give them C15 and then after 3 months see the endothelial function. And if there is improvement, I mean, that will give um, a complete validation for the benefit of C15 by itself, just to
Yeah,
see how much it will normalize endothelial function. Because what what measures the longevity and health optimization is your autonomic nervous system and endothelial function. If they are balanced and normalized, then that patient is healthy. And if it's not, then they have pathological inflammation or oxidative stress, subclinically or clinically. So have you done that endothelial function test with your supplement?
endothelial function has been assessed at the level of that biomap test I had mentioned, where it includes, and so this is in a dish, right? So diseases in a dish, in which included vascular endothelial cells, and that were mimicking a cardiovascular disease. So in those systems, it showed that it decreased pro-inflammatory cytokines, were the main effects and the main things they measured, to be honest, in those tests. So that is the extent of that. With regard to like leaky gut syndrome of gut cells, that there was a study showing it was a model of NAFLD in mice that included leaky gut syndrome, and it showed that C15 supplementation effectively closed the leaky gut and decreased, for example, the LPS levels, the leakage of bacteria into the circulation. Those are the two that I'm aware of, but nothing else
Well,
besides
it's very practically to do it because it's an FDA approved device out there, and a lot of doctors, and I can connect you with a company who
that'd be
has
great.
already doctors using it, and possibly.
Don't mention any companies. We got the CME thing.
Yeah, you got the CME. Yeah, I know. I'm not mentioning any company, but I think it's something that you may consider it.
Okay.
Great, thank you.
Yeah, it's just for clinical trials that can be done, um, and evaluate the efficacy of it. If you're interested, we can work on conducting that.
Oh, that'd be wonderful. Great to follow up.
Right.
Food-wise, sour cream, butter, or heavy cream?
Let's see if I can put it in the right order. Butter, sour cream, heavy cream, as far as C15, and I love them all. Yeah, it's a good, I think it's just a good balance. We do, again, have higher levels, much higher levels of pro-inflammatory even chain saturated fatty acids. But I think that, the lesson we're learning is what we keep learning, which is everything in moderation and don't remove everything of anything. Because we,
My grandfather died at age of 100, and he drinks a lot of goat milk. Do you think goat milk is better than cow milk?
yeah, the Sardinians have their own goats and sheep, and it's in a mountainous regions. So they make so Pecorino cheese, and I don't own any stock in Pecorino, and it's not a brand, but Pecorino cheese has 2 times higher C15 level than even cow's butter. That previously was the highest C15. So we now know how Sardinians are getting high C15 levels, but it's not just because it's from goat. It's because the goats are grass-fed, they're local, and they're feeding off of mountainous grass. So
Yeah, my dad lives in the mountains, and
ah,
that's true. So yeah, you're right.
really cool.
Question of,
I'm thinking to move to the mountain again.
uh,
Is Nevada mountains, Dr. William?
yeah, you can go to the Pocono Mountains in Northeast Pennsylvania. It's lovely there, especially in January. Take my
We
word.
actually, Nevada actually has the most mountains of any state in America.
Really?
So,
Well,
Yeah.
there you
Yes.
go.
There's no trees on them. That's the problem with the mountains.
Right.
Colorado's good, right?
But
Colorado,
yeah,
yeah.
Colorado's good too. Can we get a copy of your human studies?
Absolutely. So the first one will be the Singapore. That one is available. The as far as the clinical trials, the second one is coming. So I am not allowed to share that one. But I promise that as soon as it comes out, we'll make sure to let Dr. Bell and Dr. Melvin know.
Okay.
Dr. Steph, I posted your molecules study
Oh,
here
thank
on
you.
the chat.
Great.
We're getting a couple questions about what don't answer this one at the moment. Which pill form do you recommend? So we'll get that once we're done finished with the CME credits, and how to obtain it. Maybe wait for that. But what tests to do again? I think that was, you might have asked that, and what's the cost?
So if I'm not allowed, am I allowed to say the company name?
Not yet. Somebody asked.
Okay.
Yeah, not yet. So
I can tell you the cost of for a consumer getting the at-home test is $184 plus a $15 physician fee. For healthcare providers, that's different. But once we can talk about who provides it, then
is that a general, like, sort of omega test, or is that specific for?
It only measures C15. And so the nice thing is it provides both the percentage and the concentration, the micrograms per mil. But again, if you get a fatty acid test, if you have a fatty acid test in a lab that you like that reports percentages and has C15 in it, that works as that's great too.
All right, we'll talk about brands in a little bit, okay?
Dr. Steph, I'll model one for you. I actually have this always on my desk. So later on, once CMA is off, we're going to show off some of your stuff.
Okay, great.
The name of your website again? So
I don't think I'm allowed to say that one.
okay.
The information one, discoverc15.com.
Say it again slowly.
Discover, here, I'll put it in the, yeah. And that is not a company. That's just, so that's a place where we can put all the peer-reviewed literature that comes out. I have some catching up to do because the C15 science has been coming out pretty quickly from others these days.
In hyperferritinemia, will chelators like inositol, hexophosphate, or desoferoxamine help?
They are definitely being evaluated. for smart reasons why you asked the question. So when there have been a lot of groups looking at how to treat ferroptosis, and so they go after the mechanism. So they look for what lowers lipid peroxidation, what lowers iron deposition, and what lowers a reactive oxygen species or mitochondria repair. So they're kind of going at the pieces and parts. And so one of those are iron chelators. They it's not confirmed yet, but it is being pursued. And so for us, we were working that way with the dolphins because we recognized they were getting liver iron overload. The chelators weren't working, but phlebotomy did. So we would take 2 to 3 liters of blood off of a dolphin per week for 18 weeks, publish that work. And just like in humans, phlebotomy effectively removes iron from the liver. The problem was because we weren't fixing the problem, which were the C15 deficiencies, the iron accumulation just kind of came back over the next year. So they've moved now that they've moved to a higher C15 diet, that is effectively keeping the dolphins at bay. But if a patient is sitting on high iron overload, and I think I covered this, that C15 isn't going to treat the iron that is sitting there. But phlebotomy is very effective in removing the iron and then come in with the ability to then stop it from,
So is C15 is the iron chelation or no?
No, it just basically stops, it prevents the issue that's leading to the iron deposition.
So when you showed all the benefits, how long do you need to have been sort of supplementing with C15 before we start seeing these benefits? Is it a week? Is it 6 months? Is it 5 years?
Yeah, it's 20 years. No, I'm just kidding. So it's reliably 3 to 6 months. The 2 clinical trials were done in 3 months. In our animal models, we saw 1 to 3 months. And we didn't talk about it, but another discovery we made that we published last year in scientific reports is that we discovered, again through the dolphins, that our bodies use C15 to make a second molecule called pentadecanoyl carnitine, a metabolite. And that metabolite is the second ever discovered full-acting endocannabinoid. It fully activates CB1 and CB2 receptors. So what we're finding is when people are supplementing with C15, and I was experiencing this myself, and to be honest, I thought for sure it was a placebo effect because we didn't know about this metabolite before. But within 2 weeks, it's half of people taking C15 supplements reliably report feeling better, including deeper sleep, calmer mood, and less joint pain. And we were like, how is this even possible? And now we're understanding it's probably people who are effectively making this endocannabinoid metabolite.
So with rapamycin, at least in humans, it's been recommended that it's a intermittent dosing because you're inhibiting mTOR, and you showed that on your slides also. Is that a recommendation there? The continuous inhibition of mTOR seems to will impede immune function.
It's a great question. I think that's the difference between a druggable target where rapamycin, we aren't intended to naturally be exposed to rapamycin. It was incidentally found, and it's really good at blocking mTOR. so good that you need intermittent dosing of it. C15 is, again, a molecule that every baby gets from birth, that it is not as hard targeting with mTOR. It more does it. The best way to explain how it works is more like a balancer. So it helps trigger these mechanisms, but only to the level that's effective. And if you eat more C15, it doesn't then we don't see safety issues, but in some cases there does appear to be a U effect, which is if you get higher, it's not detrimental, but you may be losing some benefits. There really seems to be a Goldilocks zone or a sweet spot, which is what we used to eat.
Have you looked at combining C15 with rapamycin, or even any of the other mTOR inhibitors, the other, the lesser ones, metformin or quercetin, resveratrol, any of those?
We haven't, and we haven't seen studies yet combining it with other longevity, potential longevity enhancing drugs. Combo examples of studies with combo treatments are more on the cancer front, whereas 2 studies have now shown that in animal models, right, in mice, that if C15 is given with gemcitabine, that it makes gemcitabine more effective against the breast cancer tumor growth. And if C15 is given with tamoxifen in tamoxifen-resistant cancer lines, that it reverses the resistance of tamoxifen. So those are the only combo studies that we've seen to date, but I'm sure more will be coming, including rapamycin. It would make a lot of sense.
Oh,
When you
Dr.
drink a
Sapp,
lot of
that's
milk, when you drink a
okay.
lot of milk, you can lead to iron deficiency. Is that because of C15?
I don't know.
Yeah, excess of if you drink a lot of milk, especially for the babies when they drink a lot of milk or excess milk, it leads to iron deficiency. There's some chelations in the milk for the iron. So is that C15 or something else?
Yeah, that's something else because we actually have seen the opposite studies in C15 with children with iron deficiency. So, for example, there is a study done in Siberia, which showed that they looked at children with iron deficiency anemia, and they actually showed that the higher the C15 the children had in the red blood cell membranes, the less severe their anemia. So they still had iron deficiency, but it protected the cells. And a lot of iron deficiencies we've seen, and that's a whole nother talk, right? It's a relative iron deficiency. So as we take more iron so with cellular fragility syndrome, we take more iron out of the system because it's going into the tissues. And then you see a relative low iron in the blood. And so what happens is that the patients will present with what looks like iron deficiency, but they actually may have iron overload in the tissues. And there are a whole series of papers of much savvier people addressing that very sensitive topic, and recommendations for diagnostics and treatment. So yeah, no, I don't, given those studies, that does not support that C15 in milk causes iron deficiencies.
Just to address the desferoxamine question about hyperferritinemia. So this is something we monitor in our cancer patients when we do IPT. And when we see high ferritin, we have this correlation, clinical picture correlation that the cancer is more aggressive. So when we use desferoxamine, which is an iron chelator, this actually helps in relation to lowering down tumor load on our patients. So yes, we do use desferoxamine to lower down iron, and we monitor this through blood testing. So monthly ferritin and iron levels is something that we use. But again, this is a non-C15 answer, but it's just to address that hyperferritinemia question, specifically in cancer patients.
Yeah, and we have on the cancer side, we've had multiple cancer clinics contact us and saying, we're seeing patients who used to come in anemic, because of the chemotherapy, and they're not anemic, and they're patients who have always been anemic. And so then they ask them, what are they doing? And then, they say, oh, I'm supplementing with C15. And so it's enough to where, and I won't name the clinic, but one of a very famous clinic, a cancer clinic, says it is now the only type of supplementation that they allow their patients to take with cancer. So
you're saying that when you take it during cancer, it can manifest as in a blood test as they are anemic. Is that going to be a clinical picture that you expect when you take C15?
I think that very consistently, C15 has been shown to strengthen red blood cells and alleviate anemias. I think we still need to understand, it does appear that anything that results in a more fragile red blood cell, so it's not necessarily helping with producing more red blood cells, It's more about protecting against anemias that are associated with, in which you have anemia because the cells have become more fragile. So chemotherapies, anemia of aging, anemia of chronic inflammation, that those are types of anemias that have been responsive, again, in models, including dolphins.
I got two questions I'll combine. Does meat from cows have high C15, and what are good sources of C15 in the diet? I know you mentioned, milk and all, but
Yeah, so meat, yes. So ruminants make C15. So ruminants have bacteria in their gut that when they're ruminating, the bacteria in the rumen make C15, and that's how it gets into the milk, and that's how we get it. So it is in ruminant fat. So lamb and goat and cows have C15 in their fat. However, when you look at eating fat, meat fat from these animals, it does, in epidemiological studies, there does not appear to be a benefit. You're not getting C15's benefit from getting C15 from this source. And it's likely just because it just can't compete with, again, much, much, much higher levels of the pro-inflammatory fats. I think there may be a world where we're understanding ratios, because kind of like we have omega-6 to omega-3 ratios that we look at. I think there may be an even chain to like a C16 to C15 ratio, where Australian lamb has the highest C15. It still has high C16, but maybe we'll start learning that there are certain types of meats that in which we can get the good C15 without it having to compete as much.
Could it be the higher ratio of iron in a steak, for example, in comparison to a goat meat?
Oh, hey, that's a good point. Yeah, maybe it could be that now you're just adding to the problem, and yeah, not letting C15 do its job. It's like,
Right.
give me some room to work. Yeah, it's a good thought. Good thought.
But one question, I mean, it's a low-hanging fruit for me. Maybe I missed it during the presentation. Was there any baseline difference between men and women in terms of C15 level?
No, for the most part, it's pretty even, which is consistent with, again, an essential fatty acid should be, with regard to the problems of cellular fragility syndrome. If I had to guess, I would say the metabolic and fatty liver component is possibly higher prevalence in women, and that the heart component has a higher prevalence in men. But this is I can't point to any paper. It's just
Right.
Yeah.
And one quick question about what strain of bacteria produces the high C15 when we, let's say, consume inulin and other fiber?
Yeah. Okay. So we'll keep answering questions, and I will pull up that paper and put it in
Sure.
the notes.
How did, at the beginning when you told us about the dolphins and how do they keep the dolphins' records separate? How do they know that the blood you're getting from dolphin A is always from dolphin A and not from dolphin double C?
Yeah, they know these
Kind of look alike.
animals.
I hate to say it, but the pictures
Is
you show,
that speciesist? I don't know. I think that might be.
yeah, please forgive me, but how do they know that they're getting the blood from they're getting it consistently from each dolphin?
Yeah.
Don't they detect them?
Maybe.
Huh?
Don't they detect them with the radio receptors?
I'm asking. I don't know.
Yeah, they know each and every one of those dolphins intimately. And so I guess if you have a dog or a cat, and you put your dog or cat in the middle of a room filled with similar looking dogs or cats, you would know which dog is yours.
And
What's the no
Marlene,
name tags, huh?
they do tag the dolphins. The Marines and the Navy also use dolphins,
Oh, there
especially
you
in
go.
and oh yeah, and they know them by name, to say the least. And some of them they keep even in cages, big, huge cages, so they're able to control their processes and training and the whole gamut. It's pretty intense. You'd be amazed.
Yeah.
How they extract where you get it, the C15? Is it from the milk? That's how they manufacture it, or from where?
Not hypothetically, so a C15 supplement, for example, in order for it to be pharmaceutical grade and vegan friendly is plant-based C14 that adds a carbon. So that results in a greater than 99% pure C15 bioidentical molecule. And so all the studies I talked about were with that pure
From a
C15 molecule.
There's a plant.
There are plants so C14 is readily available in plants. And that includes nutmeg, coconut, others. So a common.
Okay. So by the way, Dr. Marlene Siegel is on now. She's our other
1% of those. So the
How
extraction process is.
If I drink milk, a whole milk from the mountains, how many
Oh,
cups?
maybe it's high, yeah. Then you're looking at maybe 2 to 4, maybe 2 to 3%. So it's still going to be low, but as far as eating products that come from mountainous grazing local ruminants, I would choose that as a dietary C15
How
source.
much milk to drink from the mountains? Because I have source for that. And it would
How
be
much
enough
to drink?
drink
Oh, go ahead.
of goat milk or cow milk from the mountains. That would be
Yeah, like a cup a day. Again, right, and hence that supports the Sardinian. The Sardinians actually make cheese. And so cheese is probably from mountainous grass grazing local ruminants is probably your most reliable high
Are you
C15.
from Sardinian?
Am I?
Are you from Sardinia? Are you from there?
Oh, I wish. No, I'm just trying to mimic it, and maybe we're all going to move there for Sardinians.
Yeah. What do you think, Dr. William? We moved there?
yeah, the sun shines and they got C15. I mean, what could be right? And the only other place we want to go is Easter Island so we can pick up some raw rapamycin. So and put them together, right? So
Right.
my internet went down for about a minute there, so I don't know if I missed anything or if there was it froze. So, but we should be back again. So, okay.
We just answered the meaning of life while you were
Okay.
gone.
Well, it's 42, by the way. Did you know that answer? You know what I'm talking about?
Heard that one.
Everybody know what I'm talking about? Anybody not know?
No.
Huh?
I don't.
No, there was a series of 4 books called the trilogy by a fellow named Douglas Adams in the late 60s called The Hitchhiker's Guide to the Galaxy. And it had to do with an invasion of Earth by the Vogons, which were they were the construction folks for the universe, and they went looking for the meaning of the universe, and the answer was always 42. So you can go look that up. You can go look that one up. It was on public radio. They used to have it on where I lived on Saturday nights every week. And it's back in the day it was pretty famous. The president was the guy named Zaphon. Why do I know this? Zaphon Beeblebrox. He had 2 heads and 3 heads and 2 arms, or 2 heads and 3 arms. I forget which one it is. Why do I know this?
Dr. Bill, I think it's a good way to introduce Dr. Marlene Siegel to Dr. Steph here.
That's what I was doing when my internet went down. So Dr. Siegel is our integrative veterinarian I was told you about.
Wonderful.
Dr. Venn-Watson opened by saying, it's nice that we have a veterinarian on. And I said, we had veterinarians on before. So I want to introduce you to each other. So
Hello. So nice to meet you.
so
Yeah, it was fantastic.
so
Are you familiar with parent essential fatty acids, the omega-3, omega-6 that come from like primrose oil and flax and
And they're called inherents.
they're called the parent essential
Oh,
fats.
parent.
Those that make up the cell membrane. Those are the omega-3 and omega-6 that live in the cell membrane.
Yeah, there's not as much detail, Dr. Marlene. More of from the essential fatty acid standpoint, you know, the alpha linoleic and the linolenic acids.
Yeah, those are the two I'm referring to. Yeah.
Yeah, and yeah, and in cell membranes, you know, I'm not, as far as they're polyunsaturated fatty acids, so they are susceptible to oxidation, which is why omega-3 supplements are kind of having their bad day. We do need omega-3s. They help our cell membranes be flexible, so they kind of do the opposite, right? So it's like a yin and yang relationship with regard to omega-3 fatty acids keeping our cell membranes flexible, C15 keeping them resilient. As we get older, we just become more susceptible to lipid peroxidation of those, including the LA and ALAs. So when people ask us the question about, you know, and then they may also be playing a role in, as far as omega-3s in general, and for apoptosis. But what we recommend to people is, with regard to omega-3s, is get your omega-3s from fish, if you can eat fish, or doctor prescribed prescription omega-3s. And then just be really careful about omega-3 supplements, just because these molecules are just incredibly fragile. It's not the manufacturer's fault. It's just that they're really fragile, and repeatedly now numerous studies have shown that if you pick 10 products, omega-3 fish oil supplements, 50% of them will have lipid peroxidation that are at, near, or above voluntary international safety standards. Not rancid, because that's pretty high, but high. So, and if they get to that level, it stops being effective. And because of this lipid peroxidation, half of these supplements don't have as much omega-3s in the bottle as they say, because they're starting to break down. So it's just something to be aware of. C15 is super resilient, so it's not susceptible to this in the bottle. So we're given a nice, you know, nature's advantage. But that was a long answer to your question. Yeah, we need them, and they are important in the cell membranes. But we just
Does it mean it's harmful when you say they're peroxidized, or they're just not effective? Meaning
some
adding
people,
more ROS.
yeah, some people are arguing that it's harmful. I don't know if that's true. Numerous studies have shown that people will have higher lipid peroxidation levels if they're taking, you know, supplements that are oxidized, but it kind of stops there. You know, so as far as like, does that make sense that it could be harmful? It does, but I would love to more see the study of, there aren't a lot of studies saying you take omega-3 supplements and you're harmed, unless apoptosis pans out as a cause. It's just more like they're not effective. So why do we, you know, we don't need to take, you know, 10 billion tons of fish out of the ocean if these aren't going to be effective. That's the vet side of me. We can save the fish for the dolphins.
Great. That's all I have in the chat. Anybody else have any questions? And if
Well, Dr. Bill Beverly and Miss Margaret is asking about the commercial. We're going to say
well,
that in a bit.
we're gonna end our didactic.
Well, I have some more, Dr. Bill. I think
Okay.
this is a topic that really, I think this is the first time I don't know if it's okay for Dr. Steph, because I got some questions in my email. I never had that
Yeah,
before.
go ahead.
So, Dr. Steph, you really kind of rattled the cage here and got all the people interested. So can it replace omegas? Because omegas, as we know, has SPMs, so pro-resolving mediators. And with the fatty 15, I don't know if that's something that, because as we all know, SPMs are anti-inflammatory. Does this effect also occurs with C15?
Based on our study where we went head to head against, you know, pure EPA, so not DHA, that study does support that C15 has stronger anti-inflammatory, the same plus more anti-inflammatory activities as pure EPA. What more we lean on with the omega-3. So what we tell people is if they're taking omega-3 supplements off the shelf just because they think they should, and they're otherwise healthy, as in they don't have heart disease, then it's probably not helping them. While we're still learning more and more about C15, but based upon what, the science to date, which is very consistent about C15 versus omega-3s it's kind of a mess, right? Probably because the quality of the product is resulting in a mess of scientific data. So C15 is so clean that it is more likely to consistently benefit health. But we need more clinical trials, more things need to come out. So we do argue that on that premise, it could replace C15. We are seeing more and more physicians replacing, where they're just gotten rid of C15 supplements altogether, and they've replaced it with C15 supplements, and they're seeing benefits. If a person has existing heart disease, the science does support that they may benefit even from taking off-the-shelf omega-3s under the supervision of a physician. So you're seeing like New York Times, and like this discussion is huge right now, but it is becoming very consistent that a healthy person taking omega-3 supplements likely is not going to get benefits, whether it's cognitive health, heart health. They've looked at everything, dry eye, but there's always exceptions of people. So I really think that omega-3 supplement world is falling, while prescription, pure, well-controlled grade and fish. And you did make a good comment, Beverly, about the mercury in fish. We have to be careful of that.
So we measure C-reactive proteins, and then we base whether what we're going to use as far as omega-3s and even curcumin, on those levels. Is that the C15 affect those levels also, or no, or you don't know?
It's mixed. So what's been most consistent is it lowers the pro-inflammatory cytokines IL-6, TNF-alpha, and MCP-1 across multiple species, including humans. What's less clear is CRP. Yeah, those are great ones. What's less clear is CRP. There's an epidemiological study showing that people who moved to a diet that had higher C15 had lower CRP, but it just hasn't been consistent enough, to say, oh, they have high CRP, take it to lower it. But if they had high TNF-alpha, IL-6, and MCP-1, attributed to aging, then that would be a stronger case for C15.
Great. What else you got, Dr. Melvin?
Would it be overkill when you use both omegas with C15?
No. If anything, they complement each other, and or C15 may help the lipid peroxidation.
You got a bad one, huh? It will neutralize that.
But no, it's a nice yin and yang relationship when it's working right. So you get your flexibility from your omega-3s and omega-6s, and then your rigidity and resilience from C15.
It's another curious question that, of course, we know the COVID virus is out there. It produces necrosis, pyroptosis, and maybe also accelerates spheroptosis. Is there a place for C15 in COVID, post-COVID?
Stepping carefully here, right? So no studies have been that I've seen with COVID and C15, because it's so effective and broad in an anti-inflammatory in over 18 pro-inflammatory cytokines. It would make sense that with regard to not infection with C15, but the devastating sequelae, right? So with regard to if there's ferroptosis is playing, but cytokine storms, which then we're leading right to the lung diseases and death, it would make sense that C15 may play a role in stemming the aggressive advanced form of it. But again, it's a hypothesis based upon existing data, and not any formal studies at all.
Yeah.
Coming from my
Yeah, that's a safer way to state that, right? So was this pertaining to your slide about the like a head-to-head matchup with other anti-aging remedies? Astragalus was not there, correct me if I'm wrong. And how do you compare that to astragalus? Because that's
we
probably the product for telomere lengthening. I don't know if you're aware of that.
Yeah, that we didn't go against that one. And I don't know, when there have been telomere studies involving C15, and it showed that even chain saturated fats shortened telomeres, and except for the odd chains. So the C15 and C17 had no effect. So it didn't make it longer, but it didn't make it shorter, while the even chains did. That's all I've seen on the telomere front, and we haven't put it head to head against a telomere specific compound.
Okay. You mentioned about ferritin and also, of course, as a marker to monitor iron levels. Can you also use the regular panel TIBC, iron levels, transferrin to look at ferroptosis in relation to maybe while using C15? And also, can you use that as a parameter if C15 is working on the patient?
So high ferritin with normal or close to normal transferrin saturation is a strong sign of ferroptosis and tissue iron deposition. So you can get the iron panel and then being able to see kind of like it's really the ferritin that stands out. Hyperferritinemia stands out above all else. With regard to see if C15 is working, I think it's a good measurement to say, could you have cellular fragility syndrome and ferroptosis? I think it's good for that. It's not a good marker of is C15 working, because it's telling you how much iron is deposited in the tissue. And C15, once that iron is there, C15 doesn't pull it out. So it just stops the process of what's causing the iron to be deposited in the first place. So I would actually be surprised if ferritin went down. It may be that it doesn't go up higher, but for me, I just have this like mental pressure of get that iron out and then prevent it from depositing further. And C15 the studies today do not support that C15 can actively remove existing iron from tissues once it's deposited. But let's stop the problem from happening.
Right. In dosing pathologies, do you adjust the dosing of C15 versus, of course, somebody who's taking it for anti-aging? Do you have variation in dosing?
Now that there's a widely available test, I think that could be used as a guide. Dosing. So in general, for every 100 milligrams of added C15, you increase C15 blood levels by 0.1%. So if most people are standing right on that cliff, right, of 0.2, and you want to get them more comfortable in the 0.3 to 0.4, like let's just get 100 feet from the cliff, then that 100 to 200 milligrams gets you onto safe standing. We have some of our physicians that are pushing it further to say, okay, I want to get to Sardinian level. So they're adding, the appropriate 100 milligrams doses to get themselves right now to this 0.4 to 0.6. But right now, our main goal is let's take care of the deficiencies and then work with our healthcare provider community on this longevity zone potential of C15.
You mentioned about the story of the dolphins, and they were getting their C15 from fish sources. So are there specific fish sources that we could focus on to increase C15 as part of the dietary intervention?
We could. The advantage of the dolphins, so they got it from mullet. Any type of mullet has a good amount of C15, and pinfish, which we don't eat because they're called pinfish because that you bleed when you hold them. So they have little pins all over. But the main source, the highest concentration of C15 in fish is in the skin and the heads, which of course the dolphins eat. And most cultures don't eat the skin heads. I'm half Chinese, and the person of honor at the table got the head of the fish, and now I know why.
Ah,
Hmm.
interesting. Oh, the
So,
Philippines
but
does
if
it
you
too.
but if you were to choose a fish that has the highest C15 in the meat and the muscle, it's still not, it's not the same as dairy fat, but salmon is a good choice as well.
Oh, nice. I eat on salmon.
So
Okay.
the all you can eat shrimp at the Red Lobster is not what you recommend.
Yeah, that's a good point. It's hit or miss. It's weird. Like some shrimp have a decent amount of C15 in it, and other shrimp have none, and I can't figure out why. And maybe there's so much even seasonal, different types of fish. In the winter will have higher C15 than in the summer, which makes sense because it's when they have
But
more versus less.
what
fat.
does dipping it in a cocktail sauce do to the C15?
It just makes it a more pleasant experience.
Okay, it doesn't kill it off. The question here, the C15 thin blood, the way, omegas do, increase bleeding?
No. Again, C15 as a supplement has been out in the world not as often, or not as long as omega-3s, but extensive safety studies have been done at very high doses in relevant models to specifically look at that, and there were no changes in platelets and clotting factors. So that suggests that it does not do the same as omega-3s.
Is there a maximum dose for the C15?
We haven't seen an unsafe dose. I think there's diminishing returns. So we don't push
Have you
pills.
done those like L50, is that what it's called? The L50?
Yeah, we haven't, to be honest, for us, we did generally recognize as safe, the toxicology studies to get the generally recognized as safe status for our molecule. We did it at 100 times the dose, and there was no toxicity. So we haven't found an LD50. So right now, our top recommended dose is just based upon the highest dose we dosed, not even because we saw any problems. Oh, the pancreatic enzyme deficiency. That's a good one. So absorption of C15, if you have pancreatic enzyme deficiency, if it's C15 from food, there could be a problem because C15 in food is attached to triacylglycerides, and you need those enzymes to pull off the C15 to absorb it, which is why we developed free fatty acid C15 so that it gets absorbed without needing the digestive enzymes. So it depends on source of C15.
Would the skin of the fish be better as far as mercury is concerned?
I don't know. I'm guessing. I'm guessing that mercury concentrates more in the meat, but literally I'm guessing. I haven't looked at a study that looks at mercury in skin versus.
And how long would it take to increase the C15 levels with going from 100 to 200 milligrams, and retesting?
I would give the person at least a month. At least a month.
What about dairy sensitive patients? Any
Um,
concerns?
Not for a free fatty acid supplement.
Because it's plant-based, right? You said the
Yes,
supplement.
plant-based.
Yeah, that's right. And then, okay, I think this is some, how to jack up your endurance here, but you said that it can help with hemoglobin to increase it. So can it also help with sports performance?
Possibly. Anecdotally, we hear from high performance athletes, and they say they're able to last longer and their recovery is faster. It's anecdotal. So maybe the mitochondrial component, maybe exactly the hemoglobin component, but the studies really need to be done.
Yeah, they probably want to boost their workouts, I'm assuming. So that's where it's coming from. I think it can, from what you just presented.
I mean, it makes sense. I believe them. They're the people who are very attuned to their bodies, and yeah, and it's we hear it repeatedly. So I'll just leave it at that. We try to be really careful with our anecdotal testimonials, but we do use them as hypothesis generators for formal studies.
Right.
Would you give,
And one more in the chat, Dr. Bill, I think.
would you give a person undergoing oncology support a month before retesting, or would it require more time to increase the C15 level?
I would give them, I think, I mean, if if you don't see an increase by 3 months, then something's not working, for sure. And this is across the board. The clinical trial that's about to get published, it showed everything I just said, which is for every 100 milligrams of C15, you go up, you increase by a certain amount. But there were individuals that were given 200 milligrams of C15 per day, and they did not have an increase in C15. Now, these were young adults. They were 18 to 24, and they did find that there was a lack of adherence to the protocol, and they did find that that was correlated with their C15 levels, but there was something above and beyond adherence. So I do think that there are going to be individuals that may, either from absorption standpoint, that maybe, or higher metabolism, which doesn't make a whole lot of sense, may have a harder time getting to those the stepwise level of C15 levels, which is why the testing is really important, because you need to identify. There's no point of people taking a supplement if they're not absorbing it.
I tend to more lean towards non-compliance than any of the other explanations.
That was a big one. Yeah, Jeff, who led the study, he's like, this was a really difficult population. And so, but he said he saw better adherence for this molecule or for this supplement than any other study he's done. And again, I think it might be that endocannabinoid going to work.
Okay. All right. Any more questions? Dr.
All right, I think we can, there. What would enhance absorption?
I think we're learning taking a free fatty acid form without and not as part of a high fat meal. There are 2 studies showing that in mice that when they're fed a high fat diet, their C15 levels actually go down. And so the hypothesis is that when the gut is having to choose which fats to absorb, it preferentially picks the bullies. So, the even chains, the ones that are just there in much higher prevalence.
So,
and the C15 may not make it through.
So the next question is a liposomal coating. That would be a no.
Correct. Yeah, it kind of goes against it's usually it's take with food, get the enzymes going, and so that you can absorb it. Again, it's mice, but it is interesting that, a high fat just eating, and that also goes to just eating a high fat diet, going back to high fat diets isn't, this is why we don't see all these benefits of C15 by eating a high fat diet. You've got competitors.
Does that mean there should be an empty stomach?
I think we're leaning toward that. We don't say that today, but we're trying to dive in more to Jeff's study to see if there's any way we could find out when these individuals who didn't have increases, is there any way they were like taking it with a high fat fast food meal, things like that.
Wow. Okay. That was great. Okay. Well, thank you so much.
Dr. Bill,
Another one?
we're going to let her promote it, right?
we're gonna turn off the video here.
Okay.
Microbiome health affect the C15 absorption.
Oh, that's super sweet. Microbiome health, I don't think it affects absorption. I think it affects C15 being made. So, because we now know that there are microbes, which I sent the link to which bacteria, but I think there are now 2 bacteria. Oh, Akkermansia. There's a paper that just came out showing Akkermansia makes C15, as well as the link I sent, which I've now dropped. Oh, here. Parabacteroides dystasonus is the one that uses inulin to make C.
Okay, great. Okay, well, thank you. I can't thank you enough. It's been great to have you here, and I'm frozen again. So I hope.
One slide. To be honest, it's our mission is truly how to get, now that we've with all of this science, is how do we get C15 back into people's lives, through changing agricultural practices, everything we talked about here. So let me get to the supplements. So this